Imagine a world where your medical treatment isn't just a game of probability. A
Hmm.
world where a physician doesn't just, look at a rash or evaluate a fever or glance at a scan and then just prescribe a generalized treatment based on broad population level statistics.
Right, the old trial and error approach.
Exactly.
Yeah.
I want you to picture an intervention that is custom tailored to the exact molecular instruction manual of your unique body down to the very nucleotide.
It's a fascinating thought.
It really is. And that world is, it's no longer confined to the pages of science fiction. It's the reality being engineered right now, quietly, meticulously, in computational laboratories and server farms all around the globe.
It is happening as we speak, yeah.
So welcome. Settle into whatever space you find yourself in right now. Let your shoulders drop, unclench your jaw, and just take a slow, unhurried breath.
We all need that reminder sometimes.
We really do. You are listening to Decoding the Dark, the place where we take the vast, complex, and honestly often overwhelming architecture of the modern world and distill it into something clear, calm, and deeply insightful.
It's a great space to just, well, process things.
It is. And if you find value in sharing this tranquil space with us, please remember to like and subscribe to Decoding the Dark to support our journey together.
It's a privilege to share this time. The world outside moves with such frantic urgency, but today we are going to intentionally slow down.
Absolutely. No rush today.
We are embarking on a tranquil exploration into the absolute vanguard of modern medicine. We're going to examine a vast array of sources to really understand the intricate architecture of bioinformatics, artificial intelligence, and healthcare informatics.
It's a big topic.
It is, but we'll unpack it gently. We want to understand how these fields are merging to decode the human genome, fundamentally rewrite drug discovery, and ultimately deliver highly personalized, hyper-specific clinical care.
Okay, so before we can explore how modern medicine intervenes to fix the body, we really have to thoroughly understand how it has learned to read the body's fundamental blueprints in the first place.
Right, you have to know what you're looking at before you can fix it.
Exactly. Which brings us to the origins of bioinformatics. And that term itself, it gets thrown around a lot in biotech circles.
Calls constantly.
Yeah, and it's often just treated as a synonym for, like, biology on a computer.
Right, which is a massive oversimplification.
It really is. The reality is far more expansive. Based on the material, it is this incredibly interdisciplinary field that acts as the vital bridge, combining biology, chemistry, physics, computer science, advanced mathematics, data science, and statistics.
It's a true melting pot of the hard sciences. The evolution of bioinformatics is essentially the story of how we redefined biology itself.
Redefined it how, exactly?
Well, for centuries, biology was an observational science. We looked at tissues, we categorized organs, we observed outward behavior.
Like drawing pictures of leaves in a notebook.
That's exactly, very anatomical. But bioinformatics conceptualizes biology entirely differently. It views life through the lens of physical chemistry macromolecules.
So breaking it down to the math.
Precisely. It applies rigorous mathematical techniques to organize, store, and interpret the massive data sets associated with these molecular structures. We are no longer just looking at the shape of a leaf. We are quantifying the millions of chemical interactions happening within its cells every single second.
Wow. To truly grasp the magnitude of this shift, we need to travel back to 1985.
A pivotal year.
Yeah, Robert Sintheimer, who is a biologist at the University of California, Santa Cruz, he convened a workshop proposing something that bordered on scientific heresy at the time.
Really?
He proposed mapping the entire human genetic code. And when you consider the computing power available in the mid-1980s, an era where a top-tier personal computer had perhaps 64 kilobytes of memory.
It's almost comical to think about now.
Right. The sheer audacity of proposing to sequence 3 billion base pairs of human DNA with that tech is just staggering.
It was an unimaginable proposition. They were essentially proposing to read a library of thousands of books, letter by letter, using the technological equivalent of a magnifying glass and a notepad.
That sounds agonizing.
It was incredibly slow. That effort, which formally became the Human Genome Project, took nearly 2 decades.
Wow. 2 decades?
Yeah, culminating in April of 2003. They successfully mapped our 46 chromosomes, identifying the 3 billion base pairs that contain roughly 30,000 protein-coding genes. It was routinely compared to the Apollo moon landing in terms of scientific achievement.
But the completion of that project in 2003 wasn't the medical finish line we hoped it would be.
Not at all.
Back then, there was this prevailing media narrative that once we had the map, we would immediately cure cancer, eradicate genetic diseases, and perhaps even halt aging entirely.
I remember those headlines. They were everywhere.
Yeah, that didn't happen. So I have to ask, if we had the complete hardware manual, the mapped genome, why didn't the cures immediately follow?
Because having a dictionary is vastly different from understanding a language, much less comprehending its poetry.
Oh, I like that phrasing.
In 2003, we possessed the linear sequence of letters, the A, C, T, and Gs of DNA. But we had virtually no understanding of the extraordinarily complex nonlinear interactions between those genes.
So we had the letters, but not the grammar.
Exactly. We didn't understand how the proteins they encoded actually folded, how they interacted with one another in three-dimensional space, or how environmental factors triggered their expression.
Which is crucial, right?
Absolutely vital. Most devastating illnesses, diabetes, Alzheimer's, various oncologies, they are rarely the result of a single isolated typographical error in one gene.
Right, it's really just one broken switch.
No, they are systemic failures of complex interacting networks.
So if I'm understanding this, if the genome is the static architectural blueprint sitting in a filing cabinet, we needed a way to see what the construction workers were actually building on the site.
Precisely. And that requires a transition from mere data acquisition to massive dynamic data analysis.
Because we have so much data now.
Oh, the acquisition problem has largely been solved. Next-generation sequencing, or NGS, has changed everything. It allows us to sequence entire genomes in a matter of hours for a few $100s.
Which is just a stark contrast to the billions of dollars and decades it took the original Human Genome Project.
It's night and day. NGS works by fragmenting DNA into millions of tiny pieces, attaching them to a flow cell, and sequencing them all simultaneously in parallel.
But that creates a new issue, doesn't it?
It does. NGS creates an absolute deluge of data.
Which is where bioinformatics steps in, dividing this ocean of data into specific, manageable layers, often referred to as the omics.
Right, the omics layers.
Let's walk through these slowly, because understanding precision medicine requires really understanding these layers. We start with genomics, which we've just discussed, the study of the static DNA.
That's the foundation.
Yep.
The next layer up is transcriptomics.
Transcriptomics,
Yeah.
okay.
This is the study of RNA. If DNA is the master blueprint that never leaves the nucleus, RNA represents the specific instructions that are actively being transcribed and sent out to the cellular machinery.
To build things at any given millisecond.
Yes. By sequencing the RNA, a process known as RNA-seq bioinformaticians can see exactly which genes are turned on and which are turned off in a specific tissue sample.
That sounds incredibly dynamic.
It is. It captures things like alternative splicing, where a single gene can produce multiple different variations of a protein depending on how the RNA is cut and pasted together.
So from there, we move to proteomics, which analyzes the actual proteins constructed from those RNA transcripts.
And this is where the complexity multiplies exponentially.
Because it's 3D now, right?
Exactly. A protein isn't just a linear string of amino acids.
Yeah.
Its function is entirely dictated by how it folds into intricate three-dimensional shapes.
And how it undergoes post-translational modifications, right?
Yes, like having a phosphate group attached to it, which can completely alter its behavior in the cell.
So genomics is the plan, transcriptomics is the order, proteomics is the physical machine.
That's a perfect analogy. Following proteomics is metabolomics.
Metabolomics, what does that cover?
This is the study of metabolites, which are the small molecules left behind as the chemical exhaust fumes of cellular processes.
Exhaust fumes, so it's tracking what the machine burned.
Exactly. It gives us a real-time snapshot of the physiological state of a cell. And finally, we must consider metagenomics.
Which analyzes the microbiome, right?
Yes. We are essentially walking ecosystems. The genetic material of the trillions of bacteria residing in our gut drastically influences our immune responses, our neurochemistry through the gut-brain axis, and even how we metabolize pharmaceutical drugs.
So when you stack genomics, transcriptomics, proteomics, metabolomics, and metagenomics all on top of each other, you get this rich, multi-omic profile.
A complete picture of the biological state.
And this shifts the entire paradigm of clinical treatment from empirical, symptom-based observation to mechanics-based precision.
It's a fundamental shift in how we practice medicine.
Right, we stop looking merely at the phenotype, the observable traits like elevated blood pressure or a specific tumor growth, and we start addressing the genotype, the foundational code driving the pathology.
It allows us to truly stratify patients. In the past, 10 patients with the same phenotypic diagnosis might receive the exact identical medication.
And the results would be all over the place.
Completely. 3 might be cured, 4 might see no effect, and 3 might suffer severe adverse reactions.
Because biologically, they aren't the same.
Right. Multiomics allows us to identify the specific genetic and molecular subgroups within that population, explaining why they respond differently and allowing us to tailor the intervention to the precise molecular mechanism failing in each individual.
That transition from identifying the broken mechanism to actually building a chemical to fix it brings us to drug discovery.
Which is where things get really fascinating.
We are moving from reading the biological code to utilizing artificial intelligence to invent molecular solutions.
AI is the real alchemist here.
But to really appreciate what AI is doing, we have to look at how agonizing the traditional drug discovery pipeline has been.
It is notoriously fraught. The traditional pipeline spans 10 to 15 years.
10 to 15 years just for one drug.
Yes. It begins with identifying a target, usually a protein associated with a disease. Then comes high-throughput screening, where automated robotics physically test millions of existing chemical compounds in test tubes against the target protein to see if any of them bind or react.
That sounds like an incredibly expensive game of trial and error.
It is.
And from what the sources say, those physical tests are highly prone to false positives or false negatives.
All the time.
Even if you find a compound that binds to the target in a petri dish, you then spend years testing it in animal models, only to find out it's completely toxic to the liver.
Or it can't cross the blood-brain barrier.
Right, or it simply metabolizes too quickly to be effective in a human body.
The bottleneck has always been our inability to accurately model the staggering complexity of biological systems.
Because biology is just too messy for simple math.
Exactly. Traditional statistical methods used in early computational biology relied on pre-specified parametric models. They essentially required human researchers to guess the rules of interaction beforehand.
Which is impossible given the scale of multi-omics data.
Completely impossible. Biology is profoundly non-linear. The multi-omics data we just discussed is incredibly noisy, filled with hidden relationships that no human mind and no traditional statistical model could ever discern.
Enter deep learning and machine learning architectures.
The game changers.
These AI models don't require us to pre-program the rules, do they?
No, they ingest the massive, noisy data sets and autonomously extract meaningful features, discovering the non-linear mathematical patterns hidden within.
And perhaps the most earth-shattering application of this has been in structure-based drug design.
Yes, specifically regarding the protein folding problem.
Let's slow down and unpack that because it's huge. For decades, determining the three-dimensional shape of a target protein was a monumental hurdle.
It was the holy grail of structural biology. You had to use X-ray crystallography, which involves coaxing a protein to form a crystal, a process that can take years.
And some proteins just refuse to crystallize, right?
Many do, yes.
Right.
Alternatively, you could use cryogenic electron microscopy, but both are painstaking, highly specialized physical processes.
But to design a drug like a small molecule that fits into the protein's active site like a key into a lock, you absolutely must know the exact three-dimensional topography of that lock.
You can't cut a key if you don't know the shape of the keyhole.
This is where AlphaFold completely upended the scientific world.
It really was a watershed moment.
AlphaFold didn't use physical experiments. It used a deep learning architecture, specifically attention networks, to analyze evolutionary sequence patterns.
It essentially looked at how amino acids co-evolved across different species.
Right, to predict which parts of a protein sequence must be physically close to each other in three-dimensional space, even if they are super far apart in the linear string.
The scale of its success is difficult to overstate. AlphaFold predicted the structures of over 200 million proteins.
200 million? That number is just...
Wow. To put that in perspective, before AlphaFold, the Protein Data Bank, which is the global repository of experimentally determined protein structures, held roughly 200,000 structures.
And that was accumulated over a half-century of global scientific effort.
Exactly. And then AlphaFold essentially provided the architectural blueprints for nearly every protein known to science in a matter of months.
It's breathtaking, but I want to introduce a point of friction here, just to keep us grounded.
Please do.
Having 200 million predicted structures is a monumental achievement. It gives us the shape of 200 million locks. But does possessing the exact topography of a lock guarantee that an algorithm can forge the perfect small molecule key?
And that's the multi-billion-dollar question.
Right, because protein structures aren't static blocks of concrete. The sources mention that they breathe, they fold, they shift dynamically in the cellular environment. So how does a static AI prediction account for, say, an intrinsically disordered protein that changes shape depending on what it interacts with?
That is a critical limitation, and it's one the field is actively grappling with right now. AlphaFold provides a highly accurate yet static snapshot.
Like a photograph instead of a movie.
Precisely. It does not perfectly capture the conformational dynamics, the breathing you mentioned. Furthermore, predicting the structure does not guarantee druggability.
What makes a protein undruggable?
Well, some proteins have entirely flat, featureless surfaces. They lack the deep, well-defined binding pockets that small chemical molecules need to lodge themselves into.
So there's nowhere for the key to go.
Exactly. No matter how perfectly an AI understands the shape of a featureless wall, it cannot design a key to unlock it. The biological reality remains stubbornly messy.
Even with those hurdles, though, the computational tools being deployed to design those keys are mesmerizing.
They really are.
Because once we have a target structure, we move to virtual screening.
Which replaces the robotic trial and error.
Right, instead of using robotics to physically test millions of chemicals, AI algorithms simulate the docking process. They mathematically model the atomic interactions, the van der Waals forces, the electrostatic charges, the hydrogen bonding, to predict which molecules in a digital library of billions of compounds will successfully bind to the target.
And we are no longer limited to screening libraries of existing chemicals. The real frontier of this field is de novo design, where AI invents entirely novel molecules from scratch.
Inventing new molecules, that sounds like sci-fi.
A prominent method for this utilizes generative adversarial networks, or GANs.
GANs are fascinating. Based on the reading, it's essentially two neural networks fighting each other to create something perfect.
That's a great way to visualize it. You have a generator network and a discriminator network.
Okay, so what does the generator do?
The generator's job is to create chemical structures, usually represented as strings of text called smile strings, which denote the atoms and bonds.
And the discriminator?
The discriminator's job is to look at those generated structures and compare them against a database of known, valid, safe pharmaceutical drugs.
So the generator might spit out a chemical structure, and the discriminator flags it, essentially saying, no, this carbon ring is unstable, this would be highly toxic, reject.
Exactly. And the generator learns from that rejection, tweaks the structure, and tries again.
And they play this adversarial game millions of times.
Yes. Until the generator produces a completely novel molecule that the discriminator believes is a perfectly valid, highly effective drug candidate.
To refine this further, the researchers have developed frameworks like Pac-Man,
Pac-Man, yes.
which integrates molecular structures with multi-omic data, such as gene expression profiles and protein interaction networks, to predict how sensitive a specific cancer cell line will be to a given compound.
And the evolution of that is Pac-Man NRL, which incorporates reinforcement learning.
Let's define reinforcement learning really quickly for our listeners.
In reinforcement learning, the AI agent takes actions within an environment to maximize a mathematical reward.
So like training a dog with treats, but with math.
Basically, yes. In drug design, the reward function is calculated based on several criteria: high predicted binding affinity to the target, high likelihood of being easily synthesized in a lab, and low predicted toxicity.
And Pac-Man RL is particularly revolutionary because it conditions the drug generation on a patient's specific transcriptomic profile.
It makes it profoundly personal.
Right. It isn't just designing a generic cancer drug, it is designing a molecule mathematically optimized to exploit the unique gene expression vulnerabilities of a specific patient's tumor.
It's the ultimate bespoke medicine. But while generating novel molecules is the holy grail, the economic realities of drug development are brutal.
They really are. Developing a completely new chemical entity, putting it through phase 1, 2, and 3 clinical trials, it can cost tens of millions, sometimes billions of dollars.
With a very high probability of failure at the final hurdle.
This is why AI is heavily utilized for drug repurposing, isn't it?
The financial contrast there is stark. Finding a new use for an FDA-approved drug costs a fraction of developing a new one.
The numbers in the sources are wild. It's often estimated around $8.4 million compared to $41.3 million or more for a novel entity.
Because an approved drug has already passed phase I safety trials and extensive pharmacokinetic profiling in humans, researchers can bypass years of safety testing.
And AI models excel at this by analyzing vast drug target networks. They look for hidden shared pathological mechanisms between seemingly unrelated diseases.
A great example of this happened during the height of the COVID-19 pandemic.
Right. Researchers utilized AI network-based approaches to analyze transcriptomic data.
And what they found was fascinating. They discovered shared hub genes and overlapping inflammatory pathways between severe COVID-19 infections and systemic lupus erythematosus. Yes. This allowed them to immediately identify existing lupus medications as highly viable candidates to dampen the cytokine storms seen in COVID patients.
This approach of targeting underlying molecular mechanisms, regardless of the disease's name or its location in the body, it really represents the zenith of precision medicine.
It does, and we can actually trace the conceptual roots of this back to the 1980s.
Oh, really? How so?
Well, early bioinformaticians used primitive sequence alignment tools to compare viral genomes to human genomes. They discovered a shocking sequence homology, a matching code between an oncogene in the anthraposarcoma virus and the human gene encoding platelet-derived growth factor, or PDGF.
That was a watershed moment, wasn't it?
It was. It proved that cancer could be driven by the hijacking of our own normal cellular growth factors, immediately establishing those pathways as prime targets for therapeutic intervention.
Fast forward to modern oncology, and we see the ultimate realization of that concept with drugs like bolagizumab.
Bolagizumab is a perfect example of this paradigm shift.
Historically, we categorized and treated cancers based on their anatomical origin, lung cancer, breast cancer, colon cancer.
Right.
But bolagizumab breaks that paradigm entirely. It is approved for treating solid tumors based entirely on a specific genetic signature, regardless of where the tumor is actually located in the body.
And the specific signature the drug targets is the MSIH or DMMR state.
To really understand why this is so brilliant, let's look at the mechanism. DMMR stands for deficient mismatch repair.
Right. When our cells divide and copy their DNA, the polymerase enzymes making the copy occasionally make spelling errors.
Which is normal.
Yes. Normally we have a suite of mismatch repair proteins that act as molecular spell checkers, riding along the DNA, finding those errors and fixing them.
But in DMMR tumors, those spell checkers are broken or mutated.
Exactly. As a result, the tumor's DNA accumulates thousands of uncorrected mutations. This state of hypermutation is known as microsatellite instability high, or MSIH.
Now, if you're just thinking about this logically, you might think a tumor with thousands of mutations would be stronger, right?
Sounds like it would be.
But it's actually a massive vulnerability. Because the tumor is producing highly mutated DNA, it inevitably produces highly mutated abnormal proteins.
And those abnormal proteins act as neoantigens. They look incredibly foreign to the patient's immune system.
The tumor is essentially waving a massive red flag.
Here I am, come get me.
Exactly. So drugs like bolagizumab don't attack the tumor directly. They are immune checkpoint inhibitors. They essentially release the brakes on the patient's own immune system, allowing the immune cells to recognize those highly visible neoantigens and slaughter the tumor cells.
Whether they're in the colon, the stomach, or the uterus.
It is a profound validation of the multi-omic approach.
It really is a triumph.
But designing drugs to interact with proteins or relying on the immune system is only one vector of intervention.
True.
What if the genetic blueprint itself is so fundamentally flawed that no small molecule can compensate for the resulting cascade of cellular errors?
Then you have to go to the source.
Exactly. What if, instead of designing a drug to mitigate the damage, we could open the nucleus, locate the microscopic typo in the DNA, and rewrite the broken code directly?
A permanent fix.
This marks our transition from the algorithmic design of pharmaceuticals to the staggering precision of direct genetic editing. But before we cross the threshold into the nucleus to examine these molecular tools, I want to invite you, our listener, to just pause.
Take a breath.
Let the sheer complexity of deep learning and multi-omics settle for a moment. Take a deep, relaxing breath. You are sharing this time with Decoding the Dark, a space dedicated to unraveling the intricate tapestries of our modern world with calm, unhurried curiosity.
It's important to pace ourselves with this material.
It is. If you are enjoying this tranquil exploration, please take a moment to like and subscribe to Decoding the Dark so we can continue bringing these meticulously crafted deep dives directly to you.
Now let us venture inward into the microscopic realm of genetic engineering to explore the technology that has irrevocably altered the trajectory of biological science. CRISPR-Cas9.
CRISPR is perpetually referred to in the media as molecular scissors.
I hear that constantly.
Yeah, and it's a convenient, easily digestible visual, but it severely undersells the breathtaking precision and the heavy reliance on complex bioinformatics that makes the system function in the first place.
It's much more than just a pair of scissors.
So, to move past the high school biology tropes, Let's dissect the actual biophysical mechanics of the system.
Well, the CRISPR-Cas9 system was originally discovered as an adaptive immune system in bacteria like Streptococcus pyogenes to fend off viral attacks.
Bacteria have immune systems. That alone is wild.
It is. When adapted for human genetic engineering, it consists of two primary components. First, there is the Cas9 endonuclease, the enzyme responsible for severing the DNA strands.
The scissors part.
Right. Second, there is a synthetic single guide RNA, or sgRNA.
The guide RNA is basically the programmable homing beacon. It is synthesized in a lab to perfectly match a specific sequence of 20 nucleotides in the patient's genome that we want to alter.
But Cas9 doesn't just float around the nucleus blindly checking every stretch of DNA against its RNA guide.
That would take forever.
It would be wildly inefficient across 3 billion base pairs. It requires a specific physical trigger.
And that trigger is the PAM sequence, the protospacer adjacent motif.
Exactly. For the SpeedCas9 enzyme, the PAM is a tiny 3-nucleotide sequence, specifically any nucleotide followed by 2 guanines, known as NGG.
Got NG.
Yes. The Cas9 protein rapidly scans the immense length of the DNA double helix. It is physically feeling for that NG sequence. It will not unzip the DNA, it will not expose the internal genetic code unless it docks onto that specific PAM.
Let's use an analogy. If we think of the genome as a vast, dark highway, the Cas9 is a vehicle driving at top speed, and the PAM sequence is a highly specific reflective road sign.
I like that.
It only pulls over and checks its map, the guide RNA, when it sees that exact sign. If the DNA sequence adjacent to the PAM matches the guide RNA perfectly, the Cas9 undergoes a massive conformational shift.
It engages its nucleoside domains, known as HNH and RUVC, and cleaves both strands of the DNA, creating a blunt double-strand break.
And creating the break is relatively straightforward. The actual editing relies on how the human cell panics and attempts to repair that catastrophic damage.
The cell does not like having broken DNA. It generally utilizes one of 2 highly distinct repair pathways.
The first and most common is non-homologous end joining, or NHEJ.
When the cell detects the broken strands, it acts with extreme urgency. It's essentially frantically gluing the blunt ends back together without a template.
Which is highly error-prone.
Very. It frequently results in small insertions or deletions of base pairs at the cut site, what geneticists call indels.
And these indels shift the reading frame of the gene, resulting in premature stop codons and completely disabling or knocking out the function of that specific gene.
If the goal is simply to break a dominant mutant gene causing a disease, NHEJ is sufficient. Just break it and let the cell disable it.
But if the goal is to correct a mutation, to rewrite a flawed sentence into a healthy one, the system must rely on homology-directed repair, or HDR.
HDR is elegant but difficult to trigger. In this pathway, scientists flood the nucleus with thousands of copies of a synthetic DNA template containing the correct sequence.
So the cell's repair machinery sees the break, finds the synthetic template, and essentially uses it as a tracing guide to flawlessly reconstruct the damaged section,
incorporating the new healthy code into the genome.
But the entire architecture of CRISPR is shadowed by a massive bioinformatics challenge, right?
The devastating risk of off-target effects.
Because the Cas9 enzyme is powerful, but it is not infallible. It exhibits a certain degree of mismatch tolerance.
Yes. Because the guide RNA is only 20 nucleotides long, finding a mathematically perfect match across 3 billion base pairs isn't guaranteed.
Cas9 might find a sequence that matches, say, 18 of the 20 letters, complete with a PAM sequence, and decide, close enough.
And it will cleave the DNA at that unintended site.
The consequences of an off-target cut can be catastrophic. I mean, if Cas9 accidentally cleaves an essential tumor suppressor gene, the editing process intended to cure a genetic disease could inadvertently trigger an aggressive oncology.
Ensuring safety requires mapping and predicting every single potential binding site across the entire 3-dimensional landscape of the genome.
Using CRISPR without robust bioinformatics would be analogous to performing a highly delicate microscopic neurosurgery while wearing a thick blindfold.
Guided only by vague intuition.
Exactly. We rely entirely on sophisticated computational algorithms to remove that blindfold.
And those tools are marvels of data science. Consider CRISPR.
What does that do?
It's a bioinformatics pipeline designed to analyze the raw data generated by deep sequencing the targeted DNA region post-editing. It aligns millions of sequencing reads against a reference genome to quantify the exact editing outcomes.
So it tells you how well you did.
Right. It provides researchers with precise percentages: how many cells underwent NHEJ, how many successfully utilized HDR, and the specific distribution of indel sizes.
But to prevent the off-target cuts before they ever happen, researchers use tools like CHOPSC.
It's a great name for a tool.
It is. And this algorithm doesn't just look for string matches. It employs complex thermodynamic models to evaluate the binding energy of the RNA-DNA interaction.
It scans the target exon sequences and ranks potential guide RNAs,
prioritizing those with the highest predicted on-target cleavage efficiency, while simultaneously penalizing guides that have similar sequences located elsewhere in the genome.
Another critical piece of the computational safety net is CASA-Finder.
Because the genome is so vast, searching for every possible variation of a 20-letter sequence, while allowing for 1, 2, or 3 potential mismatches, requires massive computational overhead.
Or even allowing for tiny bulges where the RNA and DNA don't perfectly align. CASA-Finder utilizes highly optimized, fast parallel algorithms to perform these exhaustive searches across the entire genome in seconds.
Highlighting every single location where the Cas9 might mistakenly bind.
Yeah.
What I find truly remarkable, though, is how the field is evolving to sidestep the dangers of double-strand breaks entirely.
Leading to next-gen CRISPR systems.
Right, like base editing and prime editing. These systems heavily rely on computational design to engineer novel fusion proteins.
Base editing is a brilliant refinement. Instead of using active scissors to cut the DNA, researchers use a catalytically dead Cas9.
A dead Cas9.
A version of the enzyme that can still find the target and bind to it, but its cutting mechanism has been completely deactivated. They fuse this dead Cas9 to a different enzyme called the deaminase.
So the dead Cas9 acts as the delivery vehicle, bringing the deaminase to the specific genetic typo.
And the deaminase then performs a precise chemical reaction on a single nucleotide.
For example, it can remove an amino group from a cytosine, converting it into a uracil, which the cell's replication machinery reads as a thymine.
You successfully change a CG base pair into a TA base pair without ever shattering the DNA double helix.
It's the difference between tearing a page out of a book and pasting a new one in versus using a microscopic pencil eraser to alter a single letter.
Prime editing pushes this paradigm even further. It fuses a partially active Cas9-1 that only nicks a single strand of the DNA rather than snapping both to a reverse transcriptase enzyme.
It utilizes an extended guide RNA that not only locates the target, but also contains the new correct genetic sequence.
The reverse transcriptase reads the RNA template and physically writes the new DNA code directly into the nicked strand.
It is the zenith of molecular word processing.
It really is.
We have spent a considerable amount of time deep in the molecular weeds today, discussing NGS sequencers reading billions of base pairs, AI architectures inventing novel therapeutics through adversarial networks, and CRISPR enzymes performing single nucleotide edits.
That's a lot of microscopic data.
But this brings us to a crucial transition. This microscopic, high-tech biological data is completely meaningless if it remains trapped in a laboratory server.
It has to reach the patient.
How does the staggering complexity of precision medicine actually reach a patient sitting nervously in an examination room? This requires us to ascend from the molecular level to the systems level, entering the domain of clinical health informatics.
This is where theoretical data must be translated into actionable clinical infrastructure. Precision medicine cannot be delivered on paper charts.
No, the foundational bedrock of this delivery system is the electronic health record, or EHR.
But the initial transition from paper to digital EHRs in the early 2000s was a fractured mess.
Hospitals bought proprietary software systems from different vendors.
Which meant a patient's oncology data in one hospital system was completely unreadable by their primary care physician system across town.
We created isolated data silos, which is fatal for precision medicine that relies on integrating a patient's entire longitudinal history.
Overcoming that fragmentation required the development of robust, vendor-neutral interoperability standards. Two of the most critical frameworks highlighted in clinical informatics are FHIR Fast Healthcare Interoperability Resources and OpenEHR.
Let's break down how these actually function, because they approach the problem differently. FHIR operates similarly to how modern websites interact.
It uses RESTful web APIs, application programming interfaces, and formats the data into standardized modular resources using JSON or XML.
So if an app needs a patient's allergy list, it sends a specific API request, and FHIR retrieves just that module, regardless of what underlying database the hospital uses.
Conversely, OpenEHR focuses on standardizing the underlying data architecture itself. It utilizes a two-level modeling approach.
It separates the static reference model, the technical structure of how data is stored, from the dynamic clinical knowledge, which is defined by clinicians using templates called archetypes.
In a truly modern health IT ecosystem, these frameworks are hybridized.
So they work together.
Yes. OpenEHR acts as the stable, semantic repository storing a complex, longitudinal clinical record, while FHIR serves as the agile transport mechanism to securely ping and share specific data points to external applications, mobile devices, or research registries.
And that seamless, standardized data layer is the absolute prerequisite for deploying clinical decision support systems, or CDSS.
A CDSS is the interface where the computational power assists the human clinician in real time.
The architecture of these systems is categorized into two distinct generations: simple CDSS and complex CDSS. Let's start with the simple ones.
Simple CDSS are foundational and ubiquitous. They operate on straightforward Boolean logic if-then statements based on established pharmacological databases and clinical guidelines.
If a physician prescribes a medication, the simple CDSS checks the patient's standardized EHR data.
If the patient has a documented allergy or is taking a conflicting medication that could cause a severe interaction, the system generates a hard stop or a warning alert.
It is a vital, transparent safety net. But the true frontier is the complex CDSS.
These systems discard simple if-then rules entirely.
They ingest the massive multi-omic profiles, the continuous sensor data, and the unstructured clinical notes, and run them through advanced machine learning algorithms, deep neural networks, and random forests.
They apply highly nonlinear mathematical models to detect subtle physiological patterns that precede clinical symptoms.
The clinical applications are staggering. Consider the deployment of deep convolutional neural networks, or CNNs, in radiology.
A complex CDSS can analyze multiparametric MRI scans to identify the morphological signatures of aggressive prostate cancer with an accuracy that often rivals or exceeds specialized human radiologists.
Or consider cardiology, where algorithms continuously analyze the complex morphology of electrocardiogram data to screen for underlying obstructive sleep apnea in patients presenting with ischemic heart disease.
Finding correlations a human would never detect from a standard ECG readout.
This predictive power is also entirely redefining telemedicine. We often think of telemedicine merely as a Zoom call with a doctor.
Right, but integrated health informatics expands this into continuous telemonitoring.
We are talking about wearable sensors transmitting real-time blood glucose variations, continuous peripheral oxygen saturation, or subtle deviations in cardiac rhythm directly to a cloud-based CDSS.
The algorithm monitors the patient 24/7, predicting and alerting the care team to an impending hypoglycemic crash or an arrhythmic event hours before the patient feels a single symptom.
Furthermore, these sophisticated AI tools enable a critical global health strategy known as task shifting,
which is so important in rural or resource-constrained environments facing severe shortages of specialized physicians.
A complex CDSS can be deployed on a tablet to assist community health workers. By inputting basic clinical data, the local worker receives real-time, evidence-based algorithmic guidance,
allowing them to safely triage and manage complex multimorbidities, like a patient suffering simultaneously from diabetes, hypertension, and a localized infection.
bringing the diagnostic capability of an urban specialist directly into the rural community.
The potential for expanding care is incredible, but
But I have to introduce a significant point of friction regarding the architecture of complex CDSS.
the notorious black box problem inherent in deep learning.
Exactly, the crisis of explainability. With a simple CDSS, the logic is entirely transparent. The doctor can see exactly which line of code triggered the allergy warning.
But a complex CDSS powered by deep neural networks relies on millions of hidden nodes, calculating obscure mathematical weights across hundreds of layers.
It is often mathematically impossible to reverse engineer the algorithm to explain how it arrived at its output.
Which is deeply problematic in medicine.
I find this deeply unsettling. If an AI checks my chart and stops a nurse from giving me a drug I'm allergic to, I feel safe. But imagine a scenario where a complex CDSS analyzes my multi-omic data and flags me for a severe, life-altering pathology requiring immediate surgery.
You ask why?
I look at my physician for reassurance and they say, I can't explain why the machine thinks you have this disease. The patterns it sees are beyond human comprehension, but statistically, the machine is rarely wrong. Doesn't that completely sever the foundational trust and the empathy that anchors the doctor-patient relationship?
You are striking at the very heart of the ethical dilemma in algorithmic medicine. When a machine's diagnostic accuracy surpasses the physician's cognitive ability to understand the machine's reasoning, The physician is demoted from an expert diagnostician to a mere interpreter of an infallible digital oracle.
The burden of trust shifts dramatically from human expertise and bedside manner to algorithmic output.
And algorithms trained on historical data can carry hidden biases. If a model is trained primarily on genomic data from populations of European descent, its predictive accuracy for a patient of African or Asian descent might plummet,
Leading to catastrophic misdiagnoses that the physician cannot mathematically interrogate.
which naturally bridges our discussion into our final domain. To train these brilliant AI systems to feed the neural networks predicting our diseases requires aggregating millions upon millions of highly intimate patient records.
Genomics, real-time sensor data, clinical histories. We have created a system of unprecedented power to heal, but simultaneously unprecedented vulnerability to exposure.
We must examine the ethical labyrinth surrounding data privacy and the legislative frameworks attempting to guard it.
The global benchmark for regulating this immense flow of biological data is the European Union's General Data Protection Regulation, the GDPR, often functioning in tandem with the Clinical Trials Regulation, or CTR.
The GDPR fundamentally shifted the power dynamic regarding digital data, declaring that the data belongs entirely to the individual, not the corporation or the hospital storing it.
And the enforcement mechanisms are draconian. Non-compliance can result in staggering penalties, reaching up to 20 million euros or 4% of a multinational corporation's total global annual turnover, whichever is higher.
Yet, despite having a unified legal framework in the EU, the actual implementation, the interpretation of how to balance data privacy against the urgent need for scientific research varies wildly between member states.
The divergence in strategy is a fascinating study in ethical philosophy. Germany, for instance, interprets the GDPR through a highly consent-heavy lens.
In the German clinical landscape, relying on broad legal exemptions for research is rare. Empirical evidence indicates that obtaining explicit, highly granular, and continuously updated consent from the patient is considered practically mandatory for sharing clinical study data.
It prioritizes the absolute autonomy of the individual over the speed of data aggregation.
Conversely, nations like France and Belgium lean heavily into the research basis exemptions provided within the GDPR. France, for example, utilizes standardized compliance frameworks, such as the MR001 methodology.
Under the MR001 framework, researchers are not forced to hunt down a patient to obtain explicit written consent every single time they want to use their data for a new retrospective study.
Instead, they operate under a model of single authorization, provided they adhere strictly to rigorous data pseudonymization protocols.
And their study design is thoroughly vetted and approved by independent ethics committees, known in France as CPPs.
It is a philosophy that attempts to balance individual privacy with a societal imperative to rapidly advance life-saving, data-intensive scientific discovery.
Regardless of whether a nation leans toward explicit consent or ethical committee oversight, all frameworks demand strict adherence to the principle of data minimization.
This principle dictates that a researcher or an AI algorithm should only collect the absolute minimum amount of personal data necessary to achieve the specific scientific goal.
It is an imperative designed to mitigate unnecessary risk, particularly when dealing with vulnerable populations. Consider a clinical trial developing an AI tool to assess motor skill development in pediatric patients.
The researchers require video footage of children performing physical tasks to train the computer vision models.
Even if the parents have signed comprehensive, informed consent documents allowing the recording, the principle of data minimization forces the researchers to ask a critical operational question. Is capturing the child's full, identifiable face strictly necessary to analyze the biomechanics of their gait?
And the answer is almost always no. The data can be gathered by applying algorithmic blurring to the faces in real time, utilizing restricted encrypted access tiers, and enforcing strict data retention limits to ensure the footage is destroyed the moment the AI is sufficiently trained.
However, the ethical landscape fractures completely when we move from phenotypic data like video to genotypic data.
The entire foundation of medical privacy relies on de-identification. Historically, if you stripped a medical file of its name, date of birth, social security number, and address, it was considered safely anonymized and could be shared freely for research.
But the anonymization of genomic data is a mathematical myth. You cannot scrub the identity from a DNA sequence because the sequence is the absolute biological definition of the identity.
Precisely. The human genome contains millions of single nucleotide polymorphisms, or SNPs points, where a single letter of DNA varies between individuals.
Computational biologists have demonstrated that you only need to match a few dozen of these specific SNPs to uniquely identify a single individual out of the entire global population.
We have seen the terrifying reality of this in practice. Researchers and law enforcement agencies have repeatedly demonstrated that they can take a completely de-identified genomic sequence from a secure clinical research database, run it against public recreational DNA databases like GEDmatch.
The platforms people use to find long-lost cousins or trace their ancestry.
Exactly. And they use basic kinship analysis algorithms to successfully re-identify the anonymous patient and, by extension, expose the genetic vulnerabilities of their entire extended family.
This creates an almost unsolvable dilemma for modern research, particularly in family-based genetic studies known as trio analyses, which involve sequencing a child and both parents to find inherited mutations.
If that data is aggregated into global federated AI training networks, guaranteeing a patient's legal right under the GDPR to be forgotten, to withdraw their consent and have their data permanently deleted, becomes a logistical impossibility.
Once the neural network has adjusted its weights based on your DNA, you cannot simply extract your contribution from the algorithm.
To attempt to navigate this, the field of clinical informatics is moving away from static, paper-based consent forms signed once in a waiting room and toward the concept of dynamic consent.
Dynamic consent utilizes interactive, secure digital patient portals, sometimes leveraging blockchain-inspired smart contracts. It transforms consent from a singular event into an ongoing, transparent relationship.
Patients can log in from their smartphones, see exactly which research studies are currently utilizing their genetic data, what the goals of the studies are, and crucially, they possess granular, real-time control to grant or revoke access to specific researchers as they see fit.
It is an elegant technological solution to a legal problem, but I return to the core, unsettling biological reality. You can blur a face in a pediatric video, you can encrypt a database, you can build a sophisticated patient portal. But how do you ever truly, fundamentally anonymize a DNA sequence?
If we look at the absolute limits of mathematics and biology, the stark truth is, you cannot. True anonymization of a full genome is an illusion. We must stop pretending the data is unidentifiable and shift our entire security paradigm.
The protection of patients cannot rely on stripping their identity. It must rely on impenetrable cryptographic security, robust federated learning models where the data never leaves the hospital server, and absolute punitive legal transparency regarding how the data is handled.
It is a delicate, high-stakes balance, managing the tension between the algorithms that could save our lives and the privacy that defines our autonomy. And that delicate balance brings us to the end of our journey today.
We have traversed an immense landscape. We started with the foundational chemical alphabet of life discovered during the Human Genome Project.
We explored the massive computational power of AI architectures designing novel therapeutics and the remarkable precision of CRISPR enzymes rewriting our very code.
We navigated the complex health IT infrastructure, the interoperable EHRs and the predictive clinical decision systems that deliver these molecular miracles to the bedside. And finally, we examined the vital yet fragile ethical frameworks attempting to guard our most intimate biological truths.
It is a breathtaking ecosystem. As we draw this deep dive to a close, I want to leave you with a mesmerizing, perhaps slightly haunting thought to ponder as you return to your day.
We touched upon the predictive power of AI and the emerging concept of digital twins. Imagine a future one that is approaching rapidly, where complex clinical decision support systems become virtually perfectly predictive.
Your digital twin, continually updated with your multi-omic data and real-time sensor telemetry, living inside a secure healthcare server, might accurately predict a severe illness, a devastating genetic breakdown, or a slow cognitive decline decades before you ever feel a single physical symptom.
When the algorithm knows your biological destiny with greater certainty than you know yourself, it fundamentally alters the trajectory of the human experience.
Exactly. If the machine tells you at age 30 that your architecture will fail at age 60, will the anxiety of knowing your exact medical timeline overshadow the incredible benefit of potentially preventing it?
How will we choose to live our lives? How will we view our own mortality when the element of biological surprise is completely removed from the human condition?
A profound question for the future of precision medicine.
Something to mull over. I want to thank you for sharing this tranquil, immersive space with us. We hope you found this exploration both deeply relaxing and intellectually illuminating. Please remember to like and subscribe to Decoding the Dark so we can continue to unravel the astonishing complexities of our world together. Until next time, take care of yourselves, breathe deep, and stay curious.